Welcome to Synapsis. I'm Gökçe Özdemir, a biomedical scientist specialized in neuroscience. In biology, synapsis is a stage during cell division where chromosome pairs line up before exchanging genetic information. And for me, synapsis is a bridge to make medical science more understandable for everyone. Thank you for being here. Let's dive in. Today, we are exploring the neuroscience of memory. Your brain is a vast, interconnected network of neurons. Each one is a microscopic powerhouse responsible for processing and storing information. Somewhere within this network, memories are formed and stored to keep you alive and functioning. From knowing how to breathe or how to walk to recalling a childhood event or just remembering where you left your keys, your brain seamlessly orchestrates countless processes. The old neuroscience painted a picture of one part of the brain doing one job and another part doing something else entirely. But now we understand that many parts of the brain can work together for a single role. And this interconnectedness holds true for memory as well. Information on a single memory can in fact be distributed across multiple brain regions. But what exactly is a memory? Is it formed by the neurons themselves, or is it more about the connections between them? To dive deeper into these questions, I'm thrilled to introduce to you a dear friend who's also a mentor, Dr. Shruti Muralidhar'ı, a neuroscientist with 20 years of experience. Shruti earned her PhD in neuroscience at EPFL in Switzerland and then completed 2 postdocs, one at the University of Utah and then another one at MIT's Pikower Institute for Learning and Memory, where she worked in the renowned Tonigawa lab. Shruti, what exactly is a memory? What makes an electrical activity between neurons turn into a memory?
So there are a lot of camps that exist in memory science, in memory neuroscience. There are groups that believe that memory is when certain number of neurons participate in a synchronous activity, and then following that, they all have the same kind of change in gene expression. That's a way of looking at it. So, they're tagged, I guess you could say, in a way. A group of neurons, let's say they do the same dance, and then they remember the dance.
It's like they make a team and they stick to a jersey color, promising that they'll always play together and they make their own unique playbook.
There you go. So that's one. And there's another group that believes, so that's a neuron centric theory, right? So in that case, you have the specific neurons that take part in specific memories. If you lose those neurons, you're going to lose those memories. That would be the logical outcome. The other sort of group of people are people who think it's not the cells that store the memory, but it's really the connection between the neurons that's where the memory is stored. And I think that also makes a lot of sense because the more you remember something consciously, the stronger your synapses get. And then the easier it gets for those synapses and the connections to fire again and again and again. So it's not so much that the memory is stored in the neurons themselves, it's really stored in the connections between the neurons. So that's another way of looking at memory. And that sort of is more flexible in the sense that you could, for example, have traumatic brain injury and lose some neurons. But that you do lose some memory, but you don't lose all of your memory. And if you keep remembering, you can remember some of the memories that you already had before and strengthen them. So your brain is a very plastic organ, and that's because neurons are very plastic cells.
I'll go back to my analogy and say that it's not just about the players, but also about the team dynamic. Even if a player is injured and can't wear the jersey anymore, someone else can step in and with enough practice, they can perform just as well to keep the memory intact. And that's really the beauty of the brain's plasticity. It finds a way to adapt and rebuild in response to new experiences and new learning or injuries.
Right. And it's not so much I think that's a great analogy because it works not just across neurons, but it also works across brain areas. So people who have like epilepsy and people who have not just traumatic brain injury, but like for example, people who have intractable epilepsy sometimes have surgeries to remove chunks of their brain, and then you're left without pieces of your brain. And they live fine. They live perfect lives, and they are creative. They don't forget stuff, and their brain just adapts. So it's a great analogy for sure.
Yeah, I think this might be the simplest way to describe what a memory is. And so far, it sounds like many regions can compensate for lost function. But when we look a level deeper, one brain structure stands out above the rest when it comes to memory. It's a star player that seems irreplaceable, and that's the hippocampus. For those who might not know, the hippocampus is a small seahorse-shaped structure located deep in the brain, tucked within the medial temporal lobe. It's known for its key role in forming new memories, particularly declarative ones, things like facts, events, and experiences. And what's really fascinating is its ancient origins. The hippocampus is one of the oldest parts of the brain in evolutionary terms. Its basic structure is conserved across many species. You'll find it in mammals, birds, and even reptiles. The fact that evolution has kept the structure relatively unchanged across such diverse species goes to show you just how vital it really is. To put this into perspective, when you compare the brains of different species, we often see significant variation in the size, the shape, and the complexity of the cerebral cortex, which is the outer gray matter that you see from the outside. And it's responsible for higher order functions like decision making and sensory processing, analytical skills in general. A little side note on the evolution of the brain before more hippocampus talk with Shruti. In humans, the cortex is highly folded, allowing for a massive surface area to be packed into our skulls. While in simpler animals like rodents, it's much smoother and less complex. Under the cortex, buried deep in the brain, are structures like the hippocampus, the hypothalamus, thalamus, amygdala, cingulate gyrus, all of which make up the limbic system that controls survival behaviors and emotional responses, reproduction, and learning. And under all of that is the oldest layer of the brain, the reptilian or the primal brain. It includes the brainstem, the cerebellum, and the basal ganglia. It's ruled by our autonomic nervous system to control our autopilot functioning, like breathing and heart rate. The term reptilian brain comes from the triune brain theory, which divided the brain into three evolutionary layers in the 1960s: the reptilian brain, the limbic system, and the neocortex. But this distinction can be a bit misleading. It implies that reptiles only have the reptilian brain with the brainstem, but no limbic system, which isn't really true. A brainstem alone can support basic survival functions like breathing, heart rate, and reflexes. However, behaviors essential for survival, such as finding food, avoiding predators, and reproduction, they all require more complex processing, which involves limbic-like structures, even in simpler animals like reptiles and fish. Modern research shows that brain evolution isn't a simple layering process where new structures are added on top of older ones. Instead, the entire brain evolves as an integrated system, with regions adapting and interacting in increasingly complex ways. Reptiles, birds, and mammals all show varying levels of limbic development, but none rely solely on a brainstem for survival. When we look at the hippocampus, it stands out as a region that's incredibly conserved in its structure and function across species. In mammals, it's really highlighted for episodic memory formation, like remembering where you parked your car. And in birds, it's vital for their spatial navigation, so that they can remember their migration routes over thousands of miles. Not too different from finding your way back to your car when you think about it. This consistency suggests that the hippocampus likely emerged hundreds of millions of years ago, well before the more complex regions of the cortex evolved. It's a testament to just how important memory and spatial navigation are for survival, whether you're a predator hunting prey or a bird finding your way back to your nest. Now that we have some evolution info under our belt, and we are clear on the importance of the hippocampus, it's a good thing that it's quite deep in the brain, not the easiest spot to injure when you hit your head. But unfortunately, even the hippocampus isn't completely safe from not getting damaged after a freaky accident. Then what would happen to our ability to form new memories?
Yeah, so unfortunately, like we said before, evolution-wise, the brain has evolved over millennia and is still evolving. And one of the side effects of that evolution is really super specialization. So even though your cortex, which is like the biggest part of your brain, is very flexible and can take multiple roles as needed, some of the very old parts of your brain don't have replacement. Unfortunately, the hippocampus is one of those where, if you don't have the hippocampus, we haven't seen it. At least there have been no observations of people who don't have hippocampus being able to form new memories. They sort of try, and there are different parts of the brain that are involved, and they do get plastic enough to try and take the place of the hippocampus, but it's never the same. So to find out that the hippocampus is responsible for memory was an absolute. There were a couple of people who had really, really traumatic accidents where the hippocampus was either destroyed or was not there to begin with, and a lot of observational studies found that these people had trouble forming memories, like they just couldn't remember. Like, for example, one of the most famous patients, people saw that even though that person had a problem with the hippocampus, he still didn't lose all of his memories, right? Like, the memories that he formed before the accident were all intact.
So, without the hippocampus, forming new memories is nearly impossible. But what's even more fascinating is that once memories are stored, they may persist, even if access to them becomes limited. That brings us to research involving Alzheimer's disease and the idea that memory itself might still be intact, just locked away. Before more details from Shruti about memories being locked away, some background on Alzheimer's. Alzheimer's is a form of dementia, and it's important to know that not all dementia is Alzheimer's. It's a progressive neurodegenerative disorder, meaning symptoms worsen over time as brain cells deteriorate and die. An early symptom is forgetfulness, can be as simple as misplacing items or forgetting recent conversations. As it progresses, communication skills also get impaired, confusion sets in, and it can become challenging to recognize even loved ones or to perform daily tasks like remembering to eat. It has two major hallmarks in the brain: amyloid plaques, which are sticky protein clumps between the neurons, and neurofibrillary tangles, which are twisted protein fibers inside the neurons. Altogether, these impair the proper functioning of the neurons and they disrupt communication between them, which ultimately leads to their death. Just like we're social creatures, our neurons are social beings. If they don't have interactions or communications, they die off. And there's not yet a cure for Alzheimer's. When you think of cells dying, this might make you think of whether stem cells could replace the dead cells in Alzheimer's. Using stem cells in the brain for various purposes is an active field of research. It's moving faster for some diseases than for others, and for Alzheimer's, unfortunately, there's still a long path ahead. For both trying to get the brain's indigenous inherent stem cell population to do something about it, and also in trying to transplant stem cells to give back function. This is a disease where the entire brain is sick, and this creates a toxic environment for new cells to come in because there's inflammation and oxidative stress, and of course the previously mentioned amyloid plaques and the protein tangles, let alone the entire issue of how difficult It is for new cells to come in to try and make new networks in a neural network that's been damaged and filled with dead cells. Imagine trying to paint a sapling in the middle of a burned down forest. Not only is the soil scorched and toxic, filled with ash and debris that hinder the sapling's growth, but the entire ecosystem it depends on, like the neighboring trees and the pollinators and the fungi, they have all disappeared or been irreparably damaged. Even if the sapling survives the toxic environment, it struggles to grow and connect because the support network that used to nourish and sustain life in the forest is gone. This is the challenge stem cells face in the Alzheimer's brain. They're trying to thrive and function in a landscape that's no longer capable of supporting them. We mentioned that a damaged hippocampus impairs new memory formation, but doesn't impair the existing memories. And this hints that the actual storage might not be in the hippocampus, but rather in the cortex. When we look at Alzheimer's disease, we run into a question of what comes first, the chicken or the egg? Is it the neurons getting sick, which then damages the connections made by them and their neighbors? Or is it a pathway damage that travels retrograde to the neuron and kills it? Derived from this, does it start in the hippocampus or does it start in the cortex and then impair a vulnerable cell population in the hippocampus? And Shruti, I don't think we will have an answer to this for many years to come. It's only one of the biggest questions in neuroscience.
It is. Absolutely. I guess what I can do is give you like an insight into how we studied Alzheimer's in the lab, and that may give you like a little counterpoint to what you were talking about. Is it, the hippocampus versus is it the cortex? Um, so since we were the lab at MIT studied memory, short-term memory versus long-term memory, it's well known that one hippocampus is the one that makes the memory, and then it sends it to the cortex, and the cortex stores all of your memories. So really, the transition between short and long-term memory happens between the hippocampus and the cortex. You were right in the sense that Alzheimer's is a disease of the entire brain. Depends on what Alzheimer's you have. Again, we can't go too much into the detail there, but there's a couple of different kinds. But almost all of them affect the entire brain. And it really, the outcomes, behavioral outcomes, the loss of memory and everything that comes with it is really because the entirety of your brain is affected. But whether Alzheimer's is really a cortical disease or a hippocampal disease, one of the insights that we had in our lab was actually really fascinating. So we used a lot of optogenetics, which is really using light to control, neuron activity. And we devised some really amazing experiments. There was a great PhD student in our lab called Biraz, and he did a bunch of these experiments, which were so elegant, and I thought were so amazing at the time. I had just joined the lab, and I was really impressed. I was really also really lucky to be able to contribute to like some aspect of that study. So the question they were asking was really like, is it the memory? Like, are memories themselves destroyed, or is it the access to the memory that's destroyed? And sort of through some really elegant experiments, they found out that it's actually the access to the memory, because they were able to access the memory in a of the genetic fashion by simply just shining light on the cells and the connections that hold the memory. And the mouse remembered, even though it was an excessively old Alzheimer's mouse. So the memories themselves in an Alzheimer's, mouse don't really go anywhere. Like you said, we may never find out whether it's like a cortical or a hippocampal or a both. There are lots of this. It's really an area of very active research right now. But I like giving this example mainly because it gives you it tells you how important it is to think a little bit outside the box.
Repeat what Shruti just said in your head. The memory itself never disappeared. Just the key to access it had gone missing. Let's dive a little deeper here. This study is based on the memory engram theory, which states that to form a memory, a group of neurons undergo biochemical and physical changes to store the newly learned information in a stable state. And when the time comes to recall the memory, they reactivate as a group, as an engram, and the memory comes back alive. Imagine you have a vintage recipe book. It's so precious that you even have a key to lock it. You have countless recipes in it, and the components of your recipes make an engram. This is where the memory for your recipe lives, in the engrams. And each recipe has a certain page to be found at. From many pages, you have to find the exact page that you need. And the question here in this study was, when you forget a recipe, is it because you lost the key to open the book to find the recipe? Or is the recipe itself incomplete or garbled? So even if you found the page, you couldn't make sense of it. To answer this, researchers used mice that showed symptoms of early Alzheimer's, so the mice couldn't access their fear memories. They were given an electric shock in a chamber, and then they forgot it happened when they were put back in the chamber, compared to healthy mice that showed fear because they remembered what happened in there before. They tagged these engram cells that are associated with the fearful experience with light-sensitive proteins. So whenever you would shine light on a specific area of their hippocampus, these cells would turn on. It's not too different from using glow-in-the-dark paint. And this is what optogenetics is, genetically modifying cells to become responsive to light. For this study, it's like turning the book to a certain page for the mice to see if there's a recipe there to work with. And long story short, by repeatedly stimulating the engram, the mice were able to use the recipe and retrieve the memory. And removing the stimulation completely removed this retrieval effect as well, confirming that the engram stimulation was absolutely required to access the recipe. The problem wasn't with the memory formation. You had written your recipe perfectly fine, but you had lost the key. I think this brings a new meaning to the general saying we have about dementia when we say someone's memory is cloudy. Maybe the sun is still there behind the clouds, so the memories might still be there, but they're just not visible because of the clouds. And perhaps potential treatments might need to be focusing on changing the weather altogether. Another key finding was that the Alzheimer's mice naturally had fewer dendritic spines, which are small buds that allow neurons to receive incoming signals from each other. Normally, when a new memory is generated, the engram cells corresponding to that memory grow new dendritic spines. But this didn't happen in the Alzheimer's mice. And this had a direct role in the sensory cue being able to stimulate the neurons to get a response from them. If the spine density does not go up during the learning process, retrieval becomes much more difficult. By optogenetically stimulating precise cells that feed into the hippocampal engram cells, they were able to increase the number of dendritic spines, which allowed the mice to retrieve the memory on their own when placed in the original chamber. This implies that memory deficits in Alzheimer's could be reduced by strengthening synapses. It just has to be done extremely precisely. Optogenetics is too invasive a method to use in humans to stimulate memories, and the closest we have to that are methods like deep brain stimulation. But that affects too much of the brain, and it's not precise enough for this particular purpose. With all this focus on memory and how important the hippocampus is, I wonder if there's a way to enhance the hippocampus. Could we make it better and faster or just more efficient? If I were to write a sci-fi book about a super memory, what could I change about the hippocampus?
We already have people in the human race who are very good at forming memories. They have surpassed, the kind of memory formation that is normally seen in human beings. But also they do it with like no specific change, like no anatomical or physical change in their hippocampus. Like all of these folks who are like super memory folks, so to the people who remember really well, people who have photographic memory, people who have adaptive memory. When you put them in a scanner and you look at their hippocampus, it doesn't look that much different. The hippocampus, it doesn't look very different from a regular person. I know there's the London taxi drivers study where they were like, well, they do have slightly larger hippocampuses, which is, it could be possible because among other things, the hippocampus is also useful in spatial memory. So reading a map to be able to orient yourself in a new place, to know where you're going, to know where you're coming from. That all of that is actually hippocampus too. And so the argument is that not only do they remember, but they actually relationally remember very well. They have lots of really good connections between their memories, so they're able to like navigate really well. I don't think adding cells to the hippocampus or adding neurons to the hippocampus or having a super hippocampus is going to do anything. I think the hippocampus by itself is a very powerful organ. The key is simply just to be able to use it well.
And maybe we are limiting our own potential by the increased use of technology for navigation, for example. Like, I personally don't go anywhere without using Google Maps. I drive on roads I've driven on, and my friends know I still get confused. And maybe that's because I'm over relying on it. And things get real sad when I start comparing myself to others, or even to animals who trace back their steps to incredible distances. And I wonder if, as humans, are we evolving to a point where it might get a bit too difficult to go back to some of the brain abilities that we used to use more?
Honestly, it's not. I mean, I feel like, again, the brain is incredibly plastic. You can do with it what you want to do with it. It's a problem of motivation. This is also not to say that you shouldn't take technology's help. I like technology's presence in our lives, and I think it has definitely enriched our lives a lot. I think the problem only comes on the over reliance. But if you live in a city and there's a part of the city you haven't been to, and you sort of know where you're going, maybe for a change, switch off your Google Maps and see how you feel about navigating by yourself. It really gives you a good idea. I mean, it's good to challenge yourself just a little bit, and it gives you a good idea of what you're capable of. And honestly, I've always found that a little bit of challenging really surprises me. I surprise myself sometimes.
The hippocampus and the whole brain itself are so powerful and adaptable that we cannot shy away from learning new things. And there's even a study that found dancing can reduce the risk of dementia because of the mental effort it takes to learn and perform every move using cognitive skills, sensory skills, spatial skills, not to mention the increased blood flow from moving around and also the release of endorphins. And this also makes me think of all the sensory cues that are part of dancing. You can remember your dance move from hearing a lyric or rhythm that reminds you where you are. Or your eye catches a detail from someone else and you remember, oh, right, it's time to turn. I need to do that. And another very powerful cue is having an emotion tied to the memory, especially when you're trying to study memory in animals, because they can't talk to you, they can't tell you what's happening, and you have to rely on their responses to emotions that you're instilling in them. And how would you go about measuring that?
Mice and rats and rodents in general have a very obvious behavioral response to fear. And you can see this, I'm sure there's tons of YouTube videos on this, but basically they freeze. It's one of the three fight, flight, or, freeze responses. And freezing is a very common response in rodents and mice and rats specifically, and we take advantage of that in the lab. So most of the memory that we study is actually fear memory in the lab because it's one of the easiest ways to understand and read. And it's very easy to see, like you would shock the mouse, you would take it out, you leave it in its cage for 2 days, but you go and put it back in the same box. It knows the box by now because it remembers the surroundings, it knows the grid, it knows that it's been here before and something bad happened. And one of the first things it will do is simply go into a corner of the box, sit there and not move. It'll freeze. And that's how we measure it. So we measure the amount of time it freezes. We measure like the strength of freezing. The more it freezes, the quicker it freezes, the better the memory is. And so that's a very easy way of measuring fear.
When we talk about fear, my mind immediately goes to the amygdala. We know it's a region that controls the fear response. And not experiencing the fear emotion in response to something dangerous, like when the amygdala is damaged, this can result in risking our lives. Fear, in this sense, it serves a critical purpose. It helps us remember the memory of danger so that we can avoid it in the future. And I guess all emotions can serve similar purposes in strengthening memories.
All strong memories in animals are formed almost always with emotions, and that's why the amygdala comes in. So any strong emotion, one will definitely become a very strong memory, but in general, emotions will help memories be formed, whether they're good memories or bad memories, whether they're good emotions or strong emotions. Memory doesn't form in air, it doesn't form by itself. Memory always forms with some sort of context. Whether, like you explained, it's a sensory context, it's a sound, it's a taste, it's a feeling, it's something you remember at the time that's not the memory itself. Or if it's emotions, it's traumatic, it's not traumatic, it's mildly traumatic, it all depends. So a good memory always has multiple components. And when I say a good memory, I just mean a memory that's strong, doesn't fade away, and stays for a long time. And it doesn't necessarily mean memory of a good thing. I guess I should reword it as a strong memory. But yeah, no, it's for sure. And contextual memory is something that's very powerful, not just for us in human beings, but even in animals in the wild. You know, a lot of animals form contextual memories. They go to a watering hole, like a zebra goes to a watering hole. It sees an alligator there for the first time. It learns to avoid the watering hole because it sees the alligator. And it doesn't have to be attacked by the alligator to remember that that's a bad place. It's already seen the alligator. So lots of context is how strong memories are.
So, to make a memory memorable, the most effective way is to match it with an emotion and context so that it's a contextual memory with multiple layers to it. We see studies about how even traumatic memories can be changed by changing the context. A strong memory for a traumatic experience can be actively negatively affecting your life. But perhaps using the brain's plasticity, you can change the context of the memory so that without changing the memory itself, you get to remember it in a different context, changing its emotional relevance.
Absolutely, yeah, and that's really the core of like therapies for PTSD these days. You know, people who come back from horrific, terrible wars have all of these memories and sounds and smells and visions are associated with these memories. But if you give them different contexts and give them context in the life of what is happening in the person now, you change basically what you're changing is the emotional valence of the memory. You're not touching the memory. Like a person who remembers getting shot will still remember getting shot, but they're not going to be scared at the sight of a gun, at like the sound of a bullet, at at something on the screen in the screen of a TV and not get like flashbacks. So really the point is to attenuate the emotional valence of the memory, which is what you want for a PTSD.
To bring back out the romantic in me, if we think of a tree with many branches, where each branch is a memory, maybe you can build new branches out of an older one that hasn't been getting enough sunlight and not giving any flowers. And that new branch you're growing, the new context to the same memory, can now grow closer to the sun and it can start flowering.
For sure, yeah. And also being aware that your brain functions in a certain way, and it's different from how everybody else's brain functions, and to be able to work with your brain rather than work against your brain. Like differences between you and me may be huge. Like I probably process trauma a lot differently than you do, but to be able to know that, to go to therapy to figure that out with somebody who knows how to do this and use the tools that you have to be able to make it easier to live with, easier to understand, easier to also, I guess, in a way, future proof yourself, right? Like we all live much longer lives than we did before, which just
effectively translates into a lot more diverse experiences, and not all of them have to be great experiences. So to be able to future proof against bad experiences, and even if you do get bad experiences, how do you process them? So honestly, I think going to therapy is like one of the best ways to do it, because most of us are not aware of the tools we already have, and going to therapy simply helps that. Like, it helps you recognize what tools.
I absolutely love this. Work with your brain rather than against it. And if you're struggling with some branches that feel heavy and rotten, trust in your brain's ability that, especially with professional help, you can learn how to prune any tree so that it can give better flowers and fruits. This organ has abilities that we're still trying to comprehend. Your brain is so good at learning new things and making new memories and building new contexts that it can even be convinced into making false memories. We have some research from Tanigawa lab at MIT where a false memory was implanted into mice. Shruti, what exactly is a false memory?
I'm going to go back to a little bit, sort of rewind a little bit about what I was talking before. Working with mice, and how do you understand whether a mouse remembers or not? Again, very elegant experiment, very simple experiment. Really, you take the mouse, you put it in box A. It has white walls and it smells of vinegar, and you shock it. You put it back in the same box the next day. You don't have to shock it; it will freeze because it remembers that it was shocked in this box. With the power of optogenetics, you find out which are these cells that make up this memory. So you can tag these cells, you can see which cells these are, and then you now take the mouse away, you put it into a completely different box. Let's say like a blue box now smells of lemon. Ideally, it should not freeze, but if you activate the memory, the fear cells in that new box, the mouse will freeze. And now, even without having been shocked in the new box, the mouse will now be scared of the new box. In our experiment, we would see the mice freezing, which is like a very reliable response to fear. But the mice wouldn't know why they were freezing. Like, they would just freeze, but there's nothing around them that says that they should be freezing. Like, they're not being shocked. They're not in the same box, and it's not the same kind of anything. So really, these are false memories. And I think this is a great demonstration of, one, the engram theory, which is, how one of the theories of how memory works in the brain, but also two, that for me, it's just the power of optogenetics. I really love it as a very elegant experiment that shows people, if you manipulate neuron activity, not in a very random sort of mass kind of a way, but really precise kind of a way, you can change precise behavior and not random behavior. Like, we didn't give the mouse epilepsy. We really just simply changed one kind of behavior, and that's freezing behavior associated with fear. It's quite really specific if you. So yeah, for me, it's a great demonstration of, what memory is, how you study it. And for me, I would think it really laid the foundation for a lot of studies that have come afterwards in terms of where memory is, how it works, how forgetting works, and how other different parts of the brain work with the hippocampus to make and unmake memory.
The key takeaway I want to leave everyone with is this. Your brain is both incredibly adaptable and deeply influenced by your experiences and emotions. This dual nature of the brain makes memory so much more than a simple record of the past. It's the foundation of who we are, shaping how we navigate our world and stay alive, and how we build our lives. Thank you everyone for joining us today. And if you enjoyed this episode, please remember to like and subscribe, and also share with others. Stay curious, stay informed, and remember to come back here next week. Santé!